Namaskar and welcome to the session on carbohydrates. This topic will be covered in two parts. The first part is on structure and physicochemical properties. So, today we will be covering the structure and physicochemical properties of carbohydrates. The objective of this session is to familiarize or reorient ourselves about the basic fundamentals of structure, classification and physicochemical properties of carbohydrates. These aspects have an impact on the processing parameters to improve the quality of the product. Let us recall that carbohydrate is one of the major food constituents which is found available in abundance on the earth. It is essentially bimolecules composed of carbon, hydrogen and oxygen, and this is produced by the photosynthetic process by the green plants using carbon dioxide, water in the presence of sunlight. Its nutritional and functional properties are like nutritional particularly, it is a source of energy, it is also the source of energy storage and also the building macromolecules like RNA, DNA also provides fiber to us which is good for the bowel regulatory movement. Then the structural integrity of the cells is also involved. The functional role, if you recall, it's about the texture, flavor and color to the product, water absorption and the gelation properties. These are the roles which it plays. So, this is one of the major food constituents which is helpful for our nutritional aspects as well as the functional role in the food processing also. So, let us start with the occurrence. Now, this is present in all the products like plant also, animal tissues and microorganisms. In plant, as you know, it is in the form of starch in stored forms and the cellulose is in the structural part as a building bricks is there. Then we have in all the fruits and vegetables, we have the simple sugars like glucose and fructose is there. In animal also the quantity is less, but it is present as in a form of glucose and it is stored as glycogen. And example of one of the from the animal sources, the lactose from the milk is also a type of a carbohydrate is there. From microorganisms, we have the xanthan gum which is available from the microorganism source. So, these are some of the sources are the examples that where the carbohydrates are present. As I mentioned, it is from the prepared from the natural source and in abundance it is available and it is present in all the things are there, like in all the fruits which what we see as a simple sugars and like the wheat or rice what we eat as a staple food is there. Now, just see that a source of the different carbohydrate source like a simple we have in the fruits, apple is there and if we have on the other range is like a wheat is there. Now, wheat contains around 71% starch is there and it also contains some other components like what we will study later, glucose, fructose, sucrose is there and the rice is around 79%. Potato has the starch around 20% and it has a cellulose 0.5%. Now, you can see the grapes have a glucose 5, fructose around 5 is there. So, that is why they are sweet is there. Apple is having the fructose 6% is there, sucrose 3% is there. So, these are the percentage of different carbohydrate components in the food. In the water percentage is given in the outside the slide is like water, wheat will have around 14%, rice has 13% water, potato has around 79%, groundnut 2%. So, this is the range. So, you can see that on one side we have the simple example of carbohydrate is sugar, and then we have the starch is there, cellulose is also there, fructose is also there, and among these the fructose is the sweetest of the carbohydrates is there. Now, see what type of carbohydrates are there. So, there are basically what we say, there are monosaccharides, disaccharides, oligosaccharides and polysaccharides. Monosaccharides mean single sugar unit, like example is glucose. Then disaccharides mean which is having a two monosaccharides, that is it sucrose, lactose, maltose, these are the example of disaccharides. Oligosaccharides, the short chain, normally from 3 to 9 monosaccharide unit is there, and polysaccharides mean long chain of monosaccharides, that is like starch, cellulose, hemicellulose and pectin. These are the examples of the types of carbohydrates are there. Now, how do we give the nomenclature name? When it was started, when the depending upon the natural sources, like if it is sugar is obtained from the cane sugar, so name sugar was given, and the beet sugar also the sugar. Now, the sugar which was obtained from malt, so it was a malt sugar, similarly grape sugar was there. Then as the time evolved, so the people started using the word the source plus in the suffix OOSC was put it, like fructose, so fruit sugar, so name was given the fructose. Maltose went from the malt plus OSC. Lactose, that is the milk sugar, the OSC is there and lactose from the milk is there. Xylose, wood sugar. So, the source plus the suffix OSC was used. Then it was evolved that is the organic compound containing carbon, hydrogen, oxygen. So, the general formula was given CXH2OY, and where so X and Y are the whole numbers, and so this was given as the hydrates of carbons, but there was no water molecule. So, the name was derived to use the saccharides is the word. Now, also more scientifically it is saccharides, and the word carbohydrates is slightly wrong, but because this is being more popular, so we using it, but it is more apt to use the saccharide word is there. Now, what is the structure of carbohydrates? First thing is they are simple linear form containing a chain of carbon atoms. So, in the subsequent slides when you will see that diagram, so it will be clear. So, it is a chain of carbon atoms, you can just imagine at this stage, and all the carbon atoms carry OH group are there except one, and one carbon atom carries a carbonyl group is also there. Now, this carbonyl group could be either an aldehyde or it could be ketone could be there, and this chain can be ranged from 3 to 100 of carbon atoms could be there, and it could be defined as polyhydroxyaldehydes or ketones are the substances which give aldehyde or ketones on breakdown. So, even on the breakdown, if it gives the aldehydes, so we use the word polyhydroxyaldehydes. If it gives the ketones, so we use the polyhydroxy ketones are there. So, this is how it is used is there. Now, this is the example of a general structure is there of the carbohydrates. You can see the first general structure is there. Then this is one is with the general structure aldehyde structure. So, is shown, and there is a one is ketone structure, and next is the fructose. So, we have a general structure, we have glucose, then we have a ketone structure, and we have a fructose. So, you can see the aldehyde group is there, and in the second it is a ketone group is there. So, this is how we show the polyhydroxyaldehyde, a general structure or with the aldehyde and one with the ketone, and with the example of glucose and fructose has been shown in this slide is there. Now, these carbohydrates can be in the linear structure and in the cyclic structure also. So, we will see that when it is in a cyclic structure, it is known as cyclic hemiacetal structure is there. The transformation from open to the cyclic structure is due to interaction between carbon at position number 1 to carbon at position number 5. This intermolecular interaction gives the cyclic hemiacetyl structure. Now, when this is obtained, we get the two forms of anomers are there. So, these are the two examples, whether we can, you can see on one side OH group is on the right side, and on one the OH group is upon the left side is there. So, this is the cyclic hemiacetyl structure is there, because it was observed that all the characteristics could not be explained because of the open chain is there. So, they like a simple example of muta rotation and optical properties could not be explained with the linear structure. So, it was observed that the number 1 and number 5 they interact is there, and during their interaction they can create in two forms are there, right, in which at the one which is called asymmetric carbon atom, there could be OH could be on the left side, and on the other OH could be on the right side could be there. Right. Just see this. So, on the polar plane, on the polarization of light, and then say, so when in alpha form the OH group will be on the below the this, and in the beta form the OH will be on the top of the is there. So, this is how the cyclic structure or what we call the pyranose structure of glucose is shown. So, now the glucose is in the linear form, in the alpha form, in the beta form during the solution is there. So, it is very important to remember that in alpha form the OH group is on the bottom, and on the beta form the OH is on the top is there. Now, how do we do the classification of this? This classification could be on the chemical structures like what we do, or the composition, or the nature of carbonyl function, or reaction with oxidizing agent, or physiological roles, but FA and WHO said that it should be done on the molecular size. So, the classification is based on the molecular size of the carbohydrates or the saccharides. So, it could be like sugars, oligosaccharides, polysaccharides, and polyols are there. Now, let us take one by one. First is the sugar group is there. Sugar, when we say, it normally comprises of monosaccharides and disaccharides. So, this is the sugar word when, so it includes monosaccharide and disaccharides is there. A single hydroxyaldehyde or ketone unit, it cannot be hydrolyzed into small units which cannot be, that is the monosaccharide is there. And the glucose and fructose are the common example of monosaccharides are there. And among these, fructose is the sweetest of all the sugar is there. Now, these are the monosaccharides, that is a simple carbohydrate, neutral, crystallizable substances, they are readily soluble in water. These are the characteristics, and it could be polyhydroxyaldehydes, aldoses, or it could be ketones are there. So, with the number 3 starts the carbon atom number. So, that then it can have 4 the tetroses, if you have the 5 pentoses, hexoses, 6 is there. So, examples are also given. If it is number 4, so examples are orthorase and thiourose. Number 5 is ribose and raffinose is there. Number 6 are example glucose, mannose, then galactose, fructose, sucrose are there. Like I mentioned in the previous, isomerism is used when we are in alpha or beta form is there, but it is the same molecular formula with different structure. That is the isomerism, if you recall your school level education or the optical properties, so you can imagine. So, this D and L configuration or alpha and beta is based on the orientation of the hydroxyl group on the asymmetric carbon is there. Just see this structure is there. So, these are the examples of monosaccharides with different number of carbohydrates. So, first is with the 3 number of carbon atoms, then we have the 4 number of carbon atoms, then next is 5 number of carbon atoms, and then 6 number of carbon atoms are there. So, this is the example of fructose is there, right. So, these are the different monosaccharides can be of 3, 4, 5 and 6 could be there. Now, these are the open chain formula, like one is the first one is the glucose is there, then is the mannose is there. Now, you see the OH group difference is there. In the fifth group, you can in the number 2, you can see the difference is there. The OH group is in the mannose is on the different side as compared to the glucose is there. Similarly, if you compare the fructose and sarbose, you can see the OH group is having different orientation is there. On the sarbose, it is on the left, and if it is on the fructose, it is on the right is there. Similarly, you can see the difference between the glucose and galactose is there in the position of OH group is there. Particularly number 4 OH group is different from the glucose group is there. So, there is a orientation of hydroxyl group and it results in stereoisomerism, and they are designated as DR left. So, they rotate the plane polarized right or left according to the word is there. So, this is the to give you more clear example of the D is there, where the it is and the one is on the left is the left orientation is there. So, these are the examples where we say that this is a D and this is the L is there. So, D series is the highest number of asymmetric carbon, OH group directed to the right, and the L series has the hydroxyl point to the left is there. Now, then we come to the, so from monosaccharides now we move to the disaccharides. So, they are made of 2 molecules of monosaccharides are there, like sucrose is an example comprising glucose and fructose, lactose comprising galactose and glucose, maltose is from the malt and it comprises 2 molecules of glucose is there. And if you see the sweetness index, sucrose has been given the 100 and then the fructose is the 173.3 is there. Then there we can also classify the disaccharides into reducing sugars and non-reducing sugars. So, reducing sugars, so they can denote electrons to other molecules, or their aldehyde and ketones have a freedom to react with others, and non-reducing sugars, they do not share the electrons like sucrose. So, the reducing glucose and maltose are the reducing sugars, whereas the sucrose is not the reducing sugar is there. Now, these are the examples. The 2 molecules of monosaccharides, they join together because of the glycosidic linkage. So, this is the showing you the glycosidic linkages for the particularly the glucose and fructose. So, they form the glycosidic linkage. Next, you see the lactose linkage is there, where the glucose and galactose are joining together. And the third type of disaccharide which is common is maltose, and where the 2 molecules of glucose are joining is there. This glycosidic linkages play a very important role in the digestion of the particularly like alpha, beta of the cellulose and other higher end of the starch are there. Now, we come to the oligosaccharides. Oligosaccharides mean where the monosaccharides units are from 3 to 9, and they are also joined by the glycosidic bonds are there. Now, they are present in plant foods like wheat, rye, vegetables, and even like onion, garlic, banana, honey is there. Now, raffinose is a trisaccharides and it is obtained in the sugar beet is there, and comprising galactose, glucose and fructose. And then the stacose is the tetrasaccharides and it is found in the pea, and it is a galactose, glucose and fructose is there. Now, this is the diagram to show that the generic that they are joined together, different monosaccharides are joined. So, they are raffinose. So, first two if you take from the left is the sucrose, then the raffinose, and then the stacose will include all the 4 monosaccharides are there. See in the next diagram. So, the first two are the cane sugar, then the beet sugar, raffinose, and in the butter, in the peas we have the stacose, and all the 4 it has been shown into the group is there. So, oligosaccharides mean 3 to 9 monosaccharide units are there. Now, what are the polysaccharides? So, the chain having more than 9 units and it could be more than 100. So, that is known as polysaccharides are there. So, there are 2 groups. Some are the chain is a linear, that is known as cellulose, glycogen. So, they are the linear chains are there. Whereas, the starch is having the branched chain is there, right. So, it is a mixture of amylose and amylopectin. Starch is having these 2 is there, and glycogen has a structure similar to that of starch, but it is shorter and has more branches. And the glycogen is it is a type of carbohydrate. In the animals, it is a form where it is stored is there, right. So, these are the examples of polysaccharides, that we have a cellulose is there, then we have a starch is there, and where the starch is made of amylase, which is linear chain. Amylopectin is a branched chain is there, and high molecular weight is there. The glycogen is more like a starch, but it is a shorter and more branched are there, but glycogen is present in the animal tissues is there. Now, these are the example, like a starch, amylose, linear glucose chain is there. Amylopectin is, you can see the branched glucose chain, which is being shown that it is a branched is there. Now, so we classify the polysaccharides as a starch and non-starch. So, starch you have seen that it comprises amylose and amylopectin, and non-starch is, we can say, comprising cellulose. Now, cellulose, the characteristics is that our digestive system cannot hydrolyze it. It is resistant to digestion is there. Hemicellulose is, they are having the low molecular weight, then the cellulose is there. Pectins are the water soluble carbohydrates, they are present in vegetables and fruits, and particularly like in papaya and these type of products it is present. Hydrocolloids are the mixed polysaccharides, they obtain from the seaweed extract, plant exudates, or even the like a microbial gum, xanthan gum. So, these fall under the hydrocolloid groups are there. Now, we come to the structural properties. Now, these carbohydrates, they contain functional groups like hydroxyl group and carbonyl group. Now, these groups contribute to hydrogen bonding and ultimately solubility in water. Like solubility of water, glucose in water is due to hydrogen bonding is there. And particularly you can see that all the carbohydrates are hydrophilic in nature because of the hydroxyl groups are there. Similarly, there are 2 words, crystalline and amorphous form, as you may be knowing also. So, crystalline is a highly ordered structure, it is like a crystalline sucrose is there. Amorphous form is a disordered structure, like a glassy state in candies is there. So, because of this, the crystalline are harder, amorphous form is always softer and more soluble is there. Now, because these structural properties have an impact on the physicochemical properties and in our further processing of fruit and vegetables. So, it is important to know. Now, what are the important physicochemical properties we will be discussing, which are in relation with the foods and the preservation, food preservation and food quality. like absorption of water, solubility, mutarotation, inversion of sugar, taste, crystallization and effect of feed, and inversion is same then inversion. So, these are some of the things we will discuss. So, first is the absorption of water. So, they are categorized as simple sugars. They are good in absorbing moisture and water. When they absorb the moisture, so they indirectly reduce the water activity and extend the shelf life, right. So, this is also one of the method of preserving also, like when we take the prepare the particularly like a juice or syrup. So, we add to the fruit juice concentrate or fruit juice, we add the sugar to this. So, it extends the shelf life is there. Now, this is one thing. So, they are hygroscopic in nature. So, if you keep your sugar crystal out, so it will get sticky is there. Now, some polysaccharides like starch, they are slow to absorb the water, but they gradually pick up the water, and when the absorption take place, but it is a reversible type of is there. And this also have the impact on the viscosity of water, like resistance to flow will be increased, and this varies with the molecular weight and concentration. And they also have the property to formation of gel, like also like when we take the starch, heat it and then cool it, it forms a gel. Or in the jam, this property is used. I mentioned pectin is one of the carbohydrate. So, we see that the right amount of pectin is present in the jam, so that it forms a gel and it traps the water which is present into this is there. So, these are the absorption of water. So, what it is good, it increases the viscosity, increases the shelf life also, it also make them hygroscopic is there. So, like you take the example of cake. So, when you want to make cake, you take sugar, so you dissolve it into the water, and then you add the other flour and other things are there. Now, then you make it. So, because it absorbs the moistures, so it gives you that it is a soft is there, and it gets the structure also because of the these properties, because it can absorb the moisture is there. Then the solubility, all the sugars dissolved in water easily is there, and this is very important in the food preparation and processing is there. Now, sugar normally will increase the boiling point of water. This we have to keep into the mind when we are preparing is there. Then the solubility varies. The fructose is the highest solubility, followed by sucrose, then glucose, then maltose, and the lactose is the least is there. Now, this solubility varies with the temperature, molecular weight and presence of the other solute. And you also know that the sugar solubility varies with the hot versus cold water. If you want to make the like any lemon drink, so if you put the sugar in cold water, it will take a long time is there. The starch is slightly insoluble in cold water, but on heating, as the starch will, the grains will swell up, they will finally mark the paste. On cooling, they come back to the same thing. They form the gel type of things is there. Then we come to the next property, mutarotation. Mutarotation is a process where the alpha and beta anomers are interconverted into in aqueous solution is there. And it has been known that the optical rotation of alpha and beta form of glucose are different. Alpha has a specific rotation of 112.2, while beta has 18.7 is there. So, equilibrium is at 52.7. So, the gradual change in specific rotation from 102 or 18.7 to 52.7 is called the mutarotation, and this interconversion between two forms, and it brings the equilibrium system containing 36% alpha and 64% beta anomer is there, and this has a role on sugar crystallization and the desired texture is there. So, this is very important in the things. This is to show you that how it takes place in a solution form, aqueous solution. So, this is the open chain structure. So, from alpha it will go to the beta and then it will go to beta to alpha, and it maintains the equilibrium level is being maintained, and it gives the desired texture to the product is there. Then the other property is the inversion of the sugar is there. The sucrose on hydrolysis with mild acid or enzyme invertase give equal amount of glucose and fructose. So, this phenomena is called inversion of sugar, and the sugar is called invert sugar. Now, this invert sugar is much sweeter than the sucrose, and it is also having the humectant properties, and that is why it is extensively used in the confectionery areas. The taste, all the sugars are sweet in taste and the degree varies from one to another. Now, the decreasing order of sweetness is, as mentioned earlier, the fructose is the sweetest, then the sucrose, then glucose, maltose and lactose. Now, this depend upon the molecular structure and receptor interactions also. So, the fructose is the sweetest and lactose is the least sweet, and the polysaccharides like starch, they are tasteless is there. Now, crystallization is, as we mentioned, it is a formation of crystal from the solution of a substance. How does it takes place? It is have a inverse relationship with the solubility. The substances which have more solubility, they are less likely to form the crystal is there. So, crystallization depends upon the nature of the solution, addition of some other ingredients in the solution, and this property is used in preparation of confection is there. In certain products, it can cause the undesirable changes, undesirable acceptability, like sweetened condensed milk in ice cream. So, you can see in ice cream you will find sometimes the crystals, in sweetened condensed milk also some crystals. So, these are lactose crystals because the solubility is low. So, they have a tendency to form the crystallization. Now, what as a processing man, what we do is we ensure that this crystallization is very fast, so that they form the small size. If the crystallization time takes a long time, then it will form the large crystals will be there. So, it is an undesirable property in certain products, but then we make it desirable by going for the fast crystallization. So, this is how, this is to show also that why these characteristics, these properties and structural property characteristics are important to know, so that we can produce the quality product or good texture product could be there. Now, with going with the fast crystallization, rapid crystallization, the low size lactose crystals will be formed, and we will not be able to judge that crystallization has taken place is there. So, these are the advantages of studying the characteristics of the carbohydrates and the physicochemical characteristics in detail, so that we can change the processing parameters effectively. Now, we come to the effect of heat. Now, heat has also affect the carbohydrates, and there are two things what we know, caramelization and Maillard reaction. Caramelization is when you heat at the melting point, you get the brown mass is there, and it mainly involves the sugar, because as we will study in the Maillard, there are two things are required, sugar and amino acids are required, while in caramelization it is only the sugar, when we heat them it takes place. So, like example of toasting of bread, roasting of rice flakes are the example, and it gives color and flavor, and the sensitive characteristics particularly in the dessert and sauces are there. And different sugars have different caramelization temperature, like galactose, glucose, they caramelize at 160, fructose at 110 and maltose at 180 degrees centigrade is there. Similarly, in the Maillard reaction, when we heat above the melting point, and this is also known as non-enzymatic browning, and it involves the aldehyde and ketone group of the sugars with the amino compounds are there, and it gives the brown color and aroma to the variety of foods like baked fruit and fried is there. Normally the reducing sugar will act are fructose, glucose, maltose and lactose, while sucrose is a non-reducing sugar is there. Now to conclude about the carbohydrates, it's a most abundant biological molecule on earth produced by photosynthesis and mainly by plants is there. It has lot of nutritional and functional importance is there, and the types, if the structure, they are the simple one, monosaccharides, we have a disaccharides, we have a oligosaccharides and polysaccharides are there. The properties which have an impact on the even the nutritional aspects and also the food processing and the quality of the products are there, like absorption of water, solubility, mutarotation, inversion of sugar. So these we covered today. Then they have the contain, these characteristics are different because of the, they have the functional group like hydroxyl group, carbonyl group, and the formation of hydrogen bond is there, which gives the solubility to this carbohydrate. So this is in the first aspect what we have covered today, structural and physicochemical properties of carbohydrates. Thank you. Thank you.